Today we are going to discuss about transposons and transposition in eukaryotes. Today's lecture is being divided into various subtopics. First, we will discuss about the discovery of transposons in eukaryotes. Then we will broadly discuss about two major types of transposons, that are the class 1 or the retrotransposon and the class 2 or the DNA transposon. Now we will go in details of the class 1 transposons and also study about the subtypes as well as the mechanism which is associated with the transposition of such transposons. Similarly, we will also discuss about the subtypes of the class 2 transposons as well as go into details of its mechanism. Now coming to the discovery of transposons, I should tell you that it was Barbara McClintock around 1980s while she was working with the variegated coat color in maize. So what she found was that there were some events where there was change or reversion of coat color, and she identified two elements. She named them as AC and DS elements, and it was found that it is the AC or the DS element which actually physically move from one location to another, and because of it, it causes changes in the coat color. Let's say when it moves from one locus to another, let's say if it's a colored locus, it will stop the gene, and because of which the kernel color will be abrogated. If it again moves out of the locus, there will be reversion to the normal phenotype. Now what happens is that such transposable elements were also found in different other eukaryotes, and today what we know that it comprises a chunk of eukaryotic genome in various organisms. For example, about 10% of the Drosophila genome is made up of transposons, and for human it varies from 30 to 35%. Now, it's very interesting to know about the types of the eukaryotic transposon present. I should mention that although Barbara McClintock was the first who discovered the transposon, but its molecular characterization was first done by Jerry Fink. So what Jerry Fink did in the experiment was that he wanted to study the HERV gene, which is associated with the amino acid histidine synthesis pathway, and he found that there were some mutants which were present, but after some time there was reversion of this phenotype. That is, it is again going back from the mutant to the wild type phenotype. And while studying the genetic details, they found that there are some eukaryotic transposon present in yeast which results in this reversion of the mutant phenotype, and they named it as the TY elements in the yeast. So from these, even from different organisms, various such elements, not the TY but the other same types of elements, were isolated, and they were broadly categorized into two classes: the class 1 or the retrotransposon and the class 2 or the DNA transposon.
Sir, we know that class 1 transposons are retrotransposons, but why are they so called?
The reason class 1 transposons are known as retrotransposon is that they have a similarity to the replication phase of the retroviruses. That is, the transposition process is it has some resemblance to the replication phase. So how do we know about this? So what are retroviruses? I will first come to it before going into it. Retroviruses are RNA viruses. They are single-stranded RNA viruses. Now the next question is, how do they infect us? Because we are DNA. So retrovirus, to infect us, they actually use that RNA strand to form a DNA. It is this DNA which gets inserted into our genome, and then again from it, it can replicate itself and form various RNA copies and then disperse. Now this the very basis of this mechanism has been utilized in retrotransposons. So we should be aware of three important genes in retroviruses, and two of them have a very important role in the transposition process of retrotransposon. The first is the GAG gene, the second is the POL gene, and the third is the ENF gene. Now all these genes are very important in the replication and the formation of the retrovirus. So what the GAG gene does is the GAG help in the RNA to mature. Now what the Pol gene does, it codes for enzymes like reverse transcriptase, it codes for protease, it codes for integrase, and it actually transcribes or it utilizes that RNA to form the DNA. Now this intermediate DNA, which is found in case of retroviruses, which can get integrated into the host, that form is known as the provirus. When the provirus gets activated, it will again form its RNA copy, and the end gene products produces the coat protein, which helps in packing of that RNA into small viral particles. Now, as I am telling you, that it is the GAG and the Pol gene which are very important in the process of transposition of these retroviruses, not the EMP gene. So during evolution, what they have done is they have got rid of the EMP gene. In most cases, only the GAG and Pol genes are present. Now, as you see in this figure, so at first, this is the entry of the package or the viral. As the virus enters, is the RNA which enters. The reverse transcriptase works and it forms the DNA, and from the DNA gets integrated into the host and it can remain in the host. Now what happens in case of the retrotransposons? In case of the retrotransposons, as you see in the next figure, they have the GAG gene and they have the Pol genes. Now this is the class 1 transposons. This GAG gene again helps maturation of the RNA, and the Pol gene reverse transcribe. So why, if you say this is important? Now these transcripts or these parts of the transposons are present in various organisms, even in their DNA form. Now these GAGs and Pols are necessary to convert it to an RNA intermediate, and then the RNA is again converted to DNA. Now what it does in this in-between phase, it replicates that copy of the transposon. So we have somewhat equivalent to copy and paste mechanism which is happening, that is replication, and it goes and sits into the new site. That is the basic essence of how the class 1 transposon works. And we actually should discuss about a very important peculiarity of the class 1 transposon, which is the presence of the long terminal repeats, and these have also some inverted repeats at its flanks. Now this is retrotransposon can actually be categorized depending on whether these LTRs are present or not. When the LTRs are present, they are known as the LTR retrotransposons, and as you see here, these are very good examples of LTR retrotransposons, and various examples of LTR retrotransposons we will go into. The first and foremost example is the TY1 elements or the TY elements in yeast. We have already discussed about the discovery of the TY elements by Jerry Fink's lab. What this figure shows you is how they get replicated or duplicated in true sense. So first, due to transcription, it will form this transcript out of this new site. This is an RNA transcript. From the RNA transcript, the proteins will be produced due to translation. These proteins will actually help utilize this one, then help make a DNA copy which will go and sit at the new site. Now depending on the position of these different genes, there can be classes, that the TY class, TY1 class or the TY3 classes. What it does, you can see here the int, which is the integrate gene, is either positioned before the reverse transcriptase in the TY1 class. In the TY3 class, this is present after the reverse transcriptase gene. Okay, now we will come to the transposition process. How was it first discovered, and how do we know that it's the TY elements which physically move from one place to another? Now a very funny experiment was performed. So what the basis of this experiment is that they attach the sequence of the TY elements in just behind the GAL4. So GAL4 is a promoter which is inducible by galactose. So now this was cloned and it was put in a bacterial genome. Now when this bacteria was fed with a source of galactose, so the GAL4 promoter will be now active and the genes will be transcribed and later translated. So as you see here, as the GAL4 promoter was activated, it produced a primary transcript. Now the transcript by itself cannot move around. the activation of the transcript happens due to splicing, because what they found by sequencing was that the size of the active mRNA was much less than that of the transcript which was earlier produced. So splicing might have happened, and then this mRNA, in this mRNA, the reverse transcriptase gene now got activated, and it again coded for the subsequent DNA, and the DNA moved to new places. And if they sequence the size of the genome, if they wanted to know the size of the genome even grossly, they found that due to transposition, the genome size was increasing. And this is the very essence of the copy and paste mechanism. We are increasing the amount of DNA present. So in TY elements, that was found. Now the next part is the question is whether these elements are only present in yeast. The answer was no. They've even screened for Drosophila, and it was Jerry Rubin's lab, who is a known Drosophila biologist. So he did some pioneering work in finding these transposable elements present in Drosophila, and he found that there are certain transposable elements which has the LTR sequence, and they also move from one place to another using RNA as an intermediate phase. So they are equivalent to retrotransposition. If you ask me, are they totally similar to the TY element? My answer is at the gene level or at the DNA level, they are not. But so PCR assays did not show that they are 100% similar. However, when they did the analysis of the amino acids which are coded by this region, they found that there are certain residues of the amino acids which are conserved both for the copia elements, which is the retrotransposon present in Drosophila, and the TY elements in yeast. Now, how do they know that these elements are present? The best studied example is the eye color mutation in Drosophila. Now, unlike us, Drosophila has red colored compound eye, and there are certain genes which actually contributes to the red color. Now, if an element go and sits in that gene, then it will, if it sits in the promoter region or if it sits in the middle, that is the exon region, then it will prevent the transcription and the translation of that sequence. So the gene will not be active. And this was the case because of the presence of the copy element moving from one place to another, the white apricot color, so they are not red. As you see, this is a wild type red eye. This is not perfectly red eye. This has a whitish. So in the white gene, the copy element was present. And if you move out the copy element from that, again you have the wild type phenotype of the white gene. So from these things, people knew that there were the retrotransposon present, and there are similarities between the copia and the TY. For example, the long terminal repeats of 276 base pair were also present, and just at the flanking of these 276 base pair, there were 17 base pair inverted repeats, and similar inverted repeats were also found in the TY elements. Is there other retrotransposons or the LTR retrotransposon present in other organisms? The answer is yes. Now, we already discussed in most cases, especially in copia as well as the TY elements, the GAG and the POL genes are present, the ENV is not present. In case of there are organisms or there are some type of transposons in organism like the gypsy or the ZAM, where the ENV gene is present, though it does not, it codes for some code protein, but it does not give rise to a total viral particle, but it gives rise to some protein which helps in the movement, which helps in other processes. Are such LTRs present in human being? The answer is yes. So sequencing found out that there are certain human endogenous long terminal repeat containing transposons present, and they are known as the HERV. Now, in most cases, the HERV is not transpositionally active. That is, they don't move around, except in 1 or 2 cases, which is the HERV-K10, where during evolution, what people have found that they sometimes move around. In case of plants, such LTR transposons are present. For example, the TA transposons in Arabidopsis, the WIS2 transposons in wheat, the TST1 transposon in potato, which are similar to the copia elements. They have the GAG and the POL genes, and their sequence similarity lies not at the DNA level, but at the amino acid level. And it has been noticed that because of the presence of gypsy or such transposable element, even the color of the flowers vary. That is, coloration variation and reversion of such variation can also happen due to the presence of transposable elements. There are sequences, especially in humans, which still use an RNA intermediate, but these long terminal repeats are not there. These are known as the non-LTR elements. For example, in humans, we know about the signs or the short interspersed nuclear element, the lines or the long interspersed nuclear elements, and as well as some composite signs present. Now coming to the lines, which is the long interspersed nuclear element. So there are regions of the human genome which have these lines, and they are mostly found in the dark 80-read region of the metaphase chromosome. If you do a chromosome sequencing or chromosome study, you will find that they are mostly located in the 80-read region. Now these, unlike the LTR transposon, codes for ORFs, and these ORFs actually code for the reverse transcriptase. Though the way the reverse transcriptase coding which happens in LTR is slightly different from what is happening in the case of the non-LTR. For example, in non-LTR, they are transcribed to RNA using the RNA Pol2 promoter which are present at the sequence at the flanking region of the lines. Now the next important elements which are there are known as the signs. We know that unlike the lines, the signs cannot move independently. The reason is that they don't code for their own reverse transcriptase. The reverse transcriptase ORF is not present in the sequence of the lines, and that's why they are so short. Now, then if you ask me, how do they move around? Now, the trick behind is that just beside somewhere in the signs are the lines, and there are at the 3' end of the sign, the sequence of the sign has some commonality to that of the line. So the reverse transcriptase which are produced by the lines can identify these 3' ends of the signs. They come and bind to this, and they can help in transposition of signs from one region to another. And as you know that because of the mobilization of the signs or even the lines during the process of evolution, there has been some improvement in some genes or the activity of the genes, or some genes has been mutated, causing some evolutionary advantage. While even when we do studies of human genome, a particular sign is very important called the ALU sequence. The ALU sequence, that stretch of sign, is identified by the restriction enzyme ALU. That's why the name is there, so it can cut at that site. And there are such sites which are present throughout the human genome. And so let's say if you take my DNA and if you take your DNA and if you do a sequencing after cutting with ALU, it won't be same because of the transposition or because of the presence of the signs are not identical as for him or me or you. So this helps us in identifying a human from another individual. As you see in this figure, the ORF of the line has ORF1, ORF2. The ORF2 codes for the polymerase, while unlike the line, the signs don't have any active ORF. We know that the lines are mostly present in the AT region. Unlike it, the signs are present in the GC-rich region unless until they are actively transposing. But before that, I should specifically tell you about what are composite signs. Now, they are not found everywhere. Now, composite sign is a peculiar case where two sign elements are present on two flanks of a gene or a particular stretch of DNA. So if the two move from one place to another, they will also take the gene from moving from one place to another. A very typical example is the bovine beta globin gene, which are present on the bovine chromosome 15. So it also have two signs. Now, because they move from one place to another, it will take the globin gene, a copy of the globin gene from one place to another. And these are peculiar example. And if you look at the figure, so the LTR retrotransposon have the GAG, have the pol. Unlike this, the lines have the GAG, have the pol, but the signs don't have. Further, the LTRs, which are the long terminal repeat having also the inverted repeats, are absent in case of the non-LTR transposons. Now, what is the basic mechanism of retrotransposition? Now, we have already covered it during discussion. Let's summarize it. So in this summary, let's take, there's a donor DNA. Okay, now this we take as a retrotransposon. Now transcription will lead to production of its corresponding RNA. Now this RNA can undergo even splicing depending on whether it is necessary or what organism it is, and then the reverse transcriptase is produced and it codes it. Because of the presence of reverse transcriptase, a DNA is produced using that RNA as a template. Now this DNA will have this flanking sequence. It will next move to a particular site, which is the target locus, and get integrated, because we know that the Pol also code for integrase, so the integration of the DNA will also occur. And I should hereby mention that don't think that the exact mechanism is same in all case of retrotransposon, but the basic mechanism of copy from a DNA to RNA and again forming a DNA is same in all of these retrotransposons. Now we shall discuss about the class 2 or the DNA transposons. The class 2 and the DNA transposons were much earlier discovered than the class 1 transposons. The first discovery and the first molecular characterization was of the P elements which are present. Then there are also the Ac-Ds elements and some other members of autonomous and non-autonomous elements. I will come very quickly on it, which are present in maize, the TAM3 family, which is present in Snapdragon, and various other which has been reported over the years. How was the P elements discovered? Now, it was a phenomenon which was happening in case of Drosophila, which lead to the discovery of P elements. This is known as hybrid dysgenesis. Hybrid dysgenesis, as I tell you, is a phenomenon where if you cross a female of a lab strain to a male which is from a natural population, then what they found is that the first filial generation was alive, but there was some issues of sterility, issues of chromosomal aberration, and this appeared again in the F2 progeny. So there was no progeny, and even if there was some progeny, it died. It was lethality. So something dramatic was happening in the body of the F1 individual, which resulted in this. Unlike this, if you just do a reciprocal cross, what I mean by that statement is if you now take the male as the lab strain and the female from the natural population, everything is fine. The F1 is fine, it's all fertile and no chromosomal aberration is seen. Now, at the molecular level, what is happening? So when they actually looked for it, they came to the conclusion that there are certain elements or certain stretches of DNA which was, according to their term, hopping around from one place to another. And these elements were called the P elements. The P element, because they are eukaryotes, has its exons and introns. Now, usually what happens, there are three major introns as you see. If two of the introns are deleted, then a 66 kilodalton transcript or protein is produced. This 66 kilodalton protein is a repressor of any transposable activity which is present in the P element. So what it means, even if the P element, the DNA is present, it will not move around because the protein come and bind and the transposase is never active. However, if three of the introns are now deleted, then the 87 kD protein is produced, which also codes for its own transposase, and hence the P element can move from one place to another. Coming to the details of the P element is, as we have discussed, in the somatic cell, because of the 66 kD protein, there is no movement of the P element. Of late, P element has been used as a vector to move genes from one region of a Drosophila genome to another region because of its transposase activity. Now, you people have the control when to activate the transposase, when not to activate the transposase. So these gives us good tool for movement of gene or for activation or deactivation of different genes. Now, coming to the next type of the element are the Ac-Ds element. Ac-Ds elements are broadly categorized into autonomous, the AC elements actually a part of autonomous and non-autonomous elements. And I should mention here the non-autonomous elements does not have its own transposes, but it requires the presence of autonomous elements in trans for its movement from one place to another. Coming to the mechanism of transposition, you can see that this is a donor site of the AC. Now, because of its transposition activity, this donor DNA is now cut. It goes to the target site. It also makes a staggered cut at the target site. It sits in the target site and then utilizes the host DNA repair mechanism to seal the gaps. And this is happening even for the DS elements, but with the help of the AC. So what we have discussed today, we have discussed about the eukaryotic transposons. We have discussed about two major subtypes, its various own subtypes, and the mechanism of transposition of the retrotransposon and the mechanism of transposition of the DNA transposon. Thank you.